First of all, thank you for the invitation. And the title is quite generalist, and there is a reason. You will find out towards the end of the talk. So I'm going to give you an overview of my facility, and then I'll just give you a little bit of my view on challenges and opportunity and how the ECHO can address some of them. So first of all, let's start. We are from Liverpool. Well, Liverpool is famous for a couple of things, like the wonderful waterfront. These guys that you may have or have not know, and they never work alone, apparently, and they have it in their DNA. So it's all about DNA. That's what we're going to talk about today. And Liverpool is also famous for one of the foundry. We have heard about the other foundries today, and we are one of the 4 foundry that's been funded by BBSRC with this huge investment for busting the synthetic biology and entering the bio design and helping the bioeconomy. So I'm also part of a bigger center. There's the Center for Genomic Research. There's a facility that offers high-throughput genomic platforms and analysis. This is a little bit of the services that are offered by our facility, and there is quite a huge variety. It goes from reading DNA to writing DNA to testing DNA in using different methodology. And this is a little bit of the platforms that we use for different application that has to do with synthetic biology or more in general with genomic or single cell. This is the GeneMill workflow. It's quite similar to any other foundry workflow. So what we do, we assemble piece of DNA, they have a function. We put together, combining with in nanoscale, we use the ECHO for that. We fabricate screening using different platforms and do QC using the PacBio. We also do some expression using microreactor, the biolector, or we can go and we have a metabolomic platform for doing some more targeted analysis. Now, we talk about challenges, opportunity. I think most of the challenges has been already mentioned in all the talks so far, so I don't come with any surprise saying that what we are trying to do is optimize, having a better reproducibility and QC all the steps of our assays. The opportunity, of course, is we want to reduce the sample size, we want to reduce the volume, because all of that means reducing costing. And also reduce risk of contamination that can be particularly relevant for some application. Now, miniaturization has been around for a while. We went from really big tube to small plates, and some of the instrument has followed up. We used to just pour our gel, agarose gel, and now we have the e-gel that's been already packed for us, and going in a scale that is a microliter scale, non-nanoliter scale, and that could be one of the challenges or opportunities for some company to be able to actually diagnosis in nanoliter scales. So, but what it really means is, like, when we reduce the cost, what we could really be is be more ambitious. We can try more. We can try to different set of conditions. We can try different samples. We can have more than triplicates. We can push that boundary a little bit. So I'll tell you a little bit of my story this summer. So I went to a Cold Spring Harbor course in synthetic biology. What they are, apart the wonderful view, what they are really famous in is this 2 weeks really intense course is a theoretical but practical course. So you are actually hand-on. And the nice thing about it is the variety of students. It comes from, in my case, there were computer scientists, they were really well-trained molecular biologists, but everything in between, and with different backgrounds. So this year was, the topic was CRISPR, TXTL, assembly, and microfluidic. Now, with quite a lot of surprise, you can read more about my story there if you're really interested in what happened, but with my really big surprise, I found that Labcyte has really kindly given an ECHO, and with the ECHO gives also this wonderful guy that is his fuzz. This is the reaction when people that don't know the ECHO see an ECHO in motion. So it's like, this is great. And when you are in this really driven environment where knowledge is there, what you need is technology. So when you combine the two, you can think in different ways, and you are exposed from different views. So what happened there, the reason because the echo was there was the simplistic one, the assembly. So the group of the assembly were just using the echo for having a modular assembly where they were trying different parts and trying to produce different compounds and screening different things. But so we were not really, let's say, allowed to use the echo, but we asked and we sort of pushed a little bit John to be a day longer than it was supposed to be. So I think he hates and loves us in different ways. So I was involved in this two module, CRISPR and TXTL. So we were thinking like, well, if a technology is there, why we cannot use in a way to test something? So, well, TXTL has been already mentioned before. This is in E. coli. So as they already, they already explained how it works. It's really simple. So they take crude extracts of E. coli, and because all the machinery is there, what they add is just a circular DNA, and then all the machinery is there, and they can produce 2 milligram per ml of protein. And Vincent Noiro and Ryan Marshall have been basically involved in preparing our TXTL mix for the course. And you could do a lot with TXTL. You can do some rapid prototype, or you could do some phage assemble, or even just minimal cell, but this is like pushing a little bit the boundary. So what we were interested in, well, first of all, we had huge problem with reproducibility among the group because, a bioinformatician has never seen a pipette before, so that means that the error rate is like just crazy. So what we were trying to do is like trying to have some knowledge of how some of our systems were working, and what we were using were a cascade system. So basically a plasmid that generates a sigma factor, and the sigma factor activate the GFP. So what we were interested is we had a quick rapid manual where we were splitting groups and we were just trying one sigma factor with several conditions, but that was really slow. It took just 2 days to find out what was which one was working better. And the additional problem was that this pipetting skills were really all over the place. So we did it with echo. We tried different things. What we tried is the single sigma factor, and then what I thought, like, what about if we took all the sigma factor together and just one GFP that was expressing one sigma factor, and see how that competes, because there must be some sort of interaction and competition between them. So that's what we found. So when you mix the sigma factor, you have different behavior. So in some cases, the mix of sigma factor produce a competitive effect. And so basically what you have is that in this case, this is with just one sigma factor, and this is all together. And as you can see, there is a reduction of GFP expression. In this case, actually, there was just one case where it was getting a little bit better. So the idea was to go back and redo this experiment and see if we could validate what we were finding and try to find a biological reason why the sigma factor were competing so massively. The other thing was about CRISPR. I know that people are, unfortunately, science is sometimes a little bit of a fashion, and now CRISPR is the big thing, so we were trying some CRISPR. But we were trying in a different way. Instead of using the cleavage ability of CRISPR, what we were doing is just using the catalytic death version of the Cas9 that basically just sits on the sequence that they recognize and then block the transcription. So in that way, what you could do is verify, for example, that that's one of the projects that Michelle Liu and Chase Baszler were working on. So they were seeing different effects of different guide RNA in expression in the GFP expression. And that was just using their standard system. They were not using TXTL. So what we were thinking as part of this project was to use the TXTL for that. So what we did, we basically did a replica of what's the experiment that was proposed in their paper, and we had basically the same results, where basically the T6 was the best in reducing the expression of GFP. We did the same with CPF1. There is a different type of CRISPR that recognizes a different size, and the guide RNA is a little bit shorter. And we did have, again, quite a lot of variation between the different guide RNA. And mostly, of course, you would expect in the one that tied to the promoter area are the one that actually cause loss of expression of that particular, of the GFP. Now what we did is we moved a little bit and we thought like, oh, shall we try something different? That's where the echo came in, and it was like really helpful a lot because we were using different conditions and was getting really difficult to take the volume down because basically we use 85% of the reaction is the crude extract of E. coli, and the rest of the reaction is the plasmid and the other components. So if you want to play a lot, you need to reduce the volume so you are able to have more condition tests at the same time. So what we test is the single strand guide RNA and the double-strand guide RNA, where the single strand basically is just an oligo with a T7 attached, and then we use the T7 DNA polymerase for elongated oligo. The reason behind that is like to reduce cost. So how you could reduce cost on buying double-stranded DNA, but just having a single-strand DNA and then make the DNA polymerase to elongate that. So we had a little bit of mixed results. At the beginning, nothing was working really. And then we start to have a little bit of better results when we added more polymerase. Still is a preliminary data. I wouldn't really go out and like shout loudly about it, but you could see that there is some repression, but it's definitely less than the double-stranded DNA. So this is an example of what happened when we did manually and when we did with echoes. Like there was like huge, the duplicates were just all over the place. So it's really useful for solving replication issue, time optimization, concentration, and volume optimization. Now I change a little bit the gear because I'm from a facility and we need to deliver some QC around plasmid. What we use now, for most of our projects, not all of them, we use some combinatory library, as has been mentioned before. The idea is that you could have this block that can be combined together, and the block has always the same overhang, so the position of the different elements in the transcription unit is always the same. But most interesting, the overhang at the end with the backbone are always the same, and you will find out why this is important. What you normally do, you build your constructs, and then the classical way of doing that, you just express in an E. coli, plate it, then take your colonies, the one that are positive, do a PCR, and then once the one that are positive, you just send it out for Sanger sequencing. This is work when your construct is small. Otherwise, if you have quite a long construct, like a 10 kb construct, what you ended up to do is design primers that goes in both directions, and for covering all your constructs. So what we were thinking of is like, we do have a lot of platforms, and we were thinking about, can we do something a little bit different? So using the fact that there are type 2 enzyme at the end, and there is an overhang that is always the same, what we could do, we could use the ARPIN adapter from the PacBio and generate what they call the smart bell. So the smart bell is basically a single-stranded circular DNA, and the way it works, the PacBio, is you got your adapter on, and then you generate the smart bell. The polymerase goes around and generate this polymerase read. That's when you trim the adapter, you get your consensus sequence. Now, the reason the part in red in the adapter is a barcode. So the idea was to take all this construct together, assuming that they are different enough for being demultiplexing, and then use the barcodes for just screening 3 colonies. So having 1 colony per barcode and pull all this plasmid together. So basically having one pot reaction when you put your construct, the enzyme, the smart bell, and ligase in one reaction. So we tried manually because the reason was it's already challenging by design, and we wanted to be sure that was working before going into further optimization. So that's what we have. This is preliminary results. A paper is on the way, so be patient. And you can see from A, B, and C that probably a paper is on the way. So the A means that I just pull all the construct together, and this is how they look like on this is an electrophoresis, a tape station, and this is basically the range of the plasmid that I put in. And the insert size was between 2.2 and 7 kb. I didn't push too much because I wanted to be sure that it was working. The B is basically after the golden gate. As you can see, it's a little bit dirty. There is a lot of noise. But if you do an exonuclease that basically get rid of all the undigested plasmid and double-stranded, the linear DNA, then that's what you get. And this is basically the fragment that you would expect due to the size of insert that you have added in. So what we were interested in is, can really the PacBio discriminate between two samples that were quite similar? The reason for that, if you are not familiar with next generation sequencing, the PacBio has quite a high error rate, but you could reduce it with coverage. So the idea was, if we can reduce that with coverage, then we could basically have a consensus and a reference, and then just align the consensus to the reference and call for SNPs. So what we do is we have done it using the onboard tools that PacBio provide in the Smart Portal, and using the Quiver. And what you could see, Quiver was able to assign the right sequence to the right constructs. So we had 65 and 66 were basically the same apart for a SNP, and Quiver was able to do that. So, and moreover, we found more little SNPs that were not in the reference, but they've been called by Quiver and confirmed by Sanger sequencing. So there is quite a nice platform, and it can be used. It can be a multiplex, probably not at the high kind of multiplexes that you could have on other platforms, but still it avoids to fragment your DNA and using later for adding adapter. So this is the idea. This is what is going to look like the workflow. We have been looking in how we can miniaturize that. So the idea is we can take our constructs and rearrange our construct on Felix. That is a standard nanoliter scale liquid handling, and then using the echo for just pulling all the plasmid together and having the highest multiplexing possible. And then the idea is just using the PacBio, but soon we have a sequel, so it should be even able to multiplex more at the same time. So if you want to hear more, just get in touch. The run should be on like in 1 or 2 weeks' time. There was a little bit of delay because we weren't able to pull. I wanted to multiplex as much as possible, so I want as much construct as possible. So I'm just hanging on a couple of constructs. So in summary, you can apply the TXTL and CRISPR and TXTL. For miniaturization and prototype, of course, you have to do a little bit of optimization, and Echo can help in that way, in giving you a variety of conditions that you can test at the same time. PacBio can be used for large construct QC, and we're thinking about using the Echo for multiplexing and to get basically as many sequences as possible at the same time, because the only downside of the next generation sequencing is the cost per run. Apart that is getting better and better in terms of accuracy. And once you have a pipeline in place, it's not too difficult to use next generation sequencing instead of Sanger sequencing. And with that, I want just to thank all these people that have been contributed at the Cold Spring Harbor course, my instructors, members of the GeneMill and the CGR, and of course Labcyte. You have been a star, and you have given us an echo for testing more. And if you want, if you want to get in touch, you're interested in any of our services, just drop an email or go and visit our website. And with that, thank you very much.